Okay students, now we have already studied about the pollination, agents of pollination, kinds of pollination. So in this session we will be studying about the post-pollination changes. After pollination, when the pollen grains fall on the stigma, what are the changes happening in the flower? So post-pollination changes we will be learning in this session.
So children, let's learn about the pollen crystal interaction, that is what happens after the pollination or post pollination condition in a flower. So here the pollen grains which are trapped by the stigma lands on the stigma. These are the pollen grains, and out of many pollen grains, all the pollen grains need not grow in the pollen tube. So it depends on the compatible kind of pollen grain only will grow the pollen tube. Rest all will stop germinating. So it depends on that stigma. It has you have studied about the genetic mechanism. So the stigma has the capability of recognizing the right kind of pollen grain, which it will help in germinating on the stigma. Other pollen grains will be degenerating. It will not lead to the germination of the pollen grain in the pollen tube. So once the pollen grain lands, the compatible one lands on the stigma, it will most of the pollen grain will start to grow the pollen tube. This is the pollen tube. And then what are the contents inside the pollen grain? You must have learnt it before in microsporogenesis. The pollen grain or the male gamete, it has this structure. Male gamete, this is the vegetative cell and this is the generative cell. The outer covering of the pollen grain, that is nothing but exine, the inner layer is the intine, and this is the germ pole. And you know that it is covered by a chemical known as sporopollenin. Here, except in this region. So this is a place through which the pollen tube will be emerging out. So it will germinate through this. So what happened? Through the germ pole, the pollen tube it germinates, and all the contents of the pollen grain, that is the vegetative nucleus and the generative nucleus, will be moved into the pollen tube. Generally, the pollen grain is the plants and all, they transfer, that is the pollen grain is released when it is in 2-cell stage. But in some cases what happened, that is when the pollen tube is germinating, it grows inside it grows from the stigma, it enters the style, and here what happened, the generative cell again divides into two to give two male gametes. So two male cells will be formed over here. So slowly by that it will enter the lower part of the ovule, and what happened at the micropylar end, this is the chalazal end and this is the micropylar end. So the pollen tube enters through the micropylar end of the ovule, and it carries the two male cells and the vegetative cell. It will help, it is rich in food supplement, it gives the nourishment to the male gametes. And also the pollen tube, it has a chemical also which will help in entering, that is to lead the part, that is the part from the style into the micropylar region. And then when it enters the micropylar region, you can look into this diagram. Here you must have already learned the structure of ovule. This is the outer integument, then you have the inner integument, this is the embryo sac. The embryo sac, as you have learned in the previous class, this is the chalazal end, this is the micropylar end. At the chalazal end, it is actually 8 nucleated but 7 cells. 8 nucleated means a chalazal end, out of the 8 cells which is formed later by mitosis, it is antipodals, 3 antipodals, and then there are 2 polar nuclei in the central cell, and here the whole thing is known as egg apparatus. Egg apparatus consists of 1 egg and 2 synergids. Okay, and synergids have thickenings at the lower end, that is known as the filiform apparatus. Filiform thickenings. This filiform thickenings will help to guide the pollen tube into the egg cell and helps in the fertilization. Now another one which we have seen is the ploidy. Whether it is a diploid in nature or what is the state of the cell